Thank you so much. Okay, so thank you so much, Ms. Wangare, for starting us off. So the first activity of the day, I would like, as we wait for a few more people to join, I would like for you guys to kindly take a poll and tell us where you're joining from. 1 minute.
I'm joining from Eldoret.
Okay, nice to meet you from Eldoret. Who else would like to share with us where they're joining from?
Hello, good morning.
Good morning.
I am joining from Zimbabwe. Thank you.
Nice to meet you from Zimbabwe.
Good morning.
Sorry, I spotlighted you. That's okay, I should have asked. Please proceed. All mute ourselves so that we stop the echo. So, Rissy, can I spotlight you or? It's okay, just proceed.
Yeah, I'm having an issue with the slides. Just one moment.
Okay. I see more people introducing themselves. Some are typing in the chat, and that's okay. Some are unmuting. We're not a big group, so feel free to tell us where you're coming from if you want to unmute and speak, or if you want to type in the chat. Maybe I can be reading.
Hello.
Effie, welcome. Where are you joining us from?
Oh, thank you. I'm Effie from Kenya. I'm joining from China.
Wonderful. Wow.
Yeah.
Today we are really, truly global. I see.
Thank you.
Okay.
Asante.
Yeah, Emmanuel Chikalipa typed a long time ago. He said, the University of Zambia, research interests, plant breeding, genomics, and seed systems. Then we have Tom Boya Nyangwara, University of Nairobi. Winnie Wanjiko pursuing masters in UCAS, China, specializing in plant genetics, botany. Due to the epidemic, I have suspended the studies while here in Kenya. And Tom Nyangwara adds his research interests are mental health and chronic diseases. Thomas Mawora from Maseno, Kenya. Wonderful. And Rissy, you may now proceed. I see you've shared a link that we can now open. There's somebody else who wants to tell us? Hello, welcome. Hello, we can hear you. Was it Austin Muswere who had unmuted or who was talking? All right.
Hello.
Hello. Yes, we can hear you.
And I'm from Africa University, Zimbabwe. Thank you.
Africa University, Zimbabwe. Thank you so much, Austin. So I think now hand over back to Rissy for the poll.
Yes. So I have just shared with you guys a link and it will take you to Slido. For today's session, we will be using it to answer some polls. So you can also just use it, you click it on your iPhone, whichever device you're using, and then you can also get the first poll is just running at the moment. It's asking you where you're joining from, so you can post it there and we'll be able to see like majority of the individuals where they're joining from. So let's try that, and I also would love to know whether it's working on your end or not. Yes, I'm seeing Zimbabwe, Kenya. Where else? Which country are you representing? Kenya again. Kenya, Nanyuki. Oh, nice. Any other person? All right. So that poll will keep on running, and as more people keep on joining, you can continue to send us your location. Oh, I'm seeing Zambia. Oh, that's nice. Nairobi, Uganda. Okay. Eldoret. Oh, Eldoret people in the house. Hello. Okay, so we are going to move now into the next poll, whereby I would like you to type in the slide as well, another poll. Just let me know in one word.
Could you speak up a little bit, Rissy?
Right.
Could you add your volume or come closer to your gadget or wear earphones so that we can get your sound loud and clear? I mean, it's clear, but the volume is a little low.
All right.
So if you could speak a little louder, that would work. Thank you.
Okay, Wangari, I hope I won't be shouting. Okay, let's try with the earphones. How is it now?
A little better. Can everybody hear Rissy okay?
Yes.
Okay, good. So I think we can
Alright.
proceed. Thank you.
All right, so I'm saying that the next poll, I would like you to describe in one word, what are your expectations? Like, what do you expect to learn from this session? Okay, so let's see. Let's see what people are thinking in one word.
I think if you're on a computer, you need to move from the tab where the Zoom is back to the slider for the next question. So sometimes people think it types right into the Zoom. So you need to remember that.
Yeah,
go to the slider link to answer the second question. Thank you.
All right, somebody is saying methods. Thank you so much for your response. What is a genome? All right, that's interesting. Human genome, great, great. Why genome sequencing? Okay, all right. I hope our panelists are noting this down. Okay, one more. What is genome sequencing? Great, great. What is genome sequencing again? Human genome? Genome sequencing. The benefits of HGS, all right. Okay, so you can keep on sending in your expectations. I'll be monitoring the tab, and we will see at the end of the sessions whether those expectations were met. So just to get everybody at bar with what we are discussing today, for those who are new in the field, I will start with a brief introductory session. So from our topic today, which says, would you allow for your genome to be sequenced? I know at this point in time, it seems like a very ambiguous question whereby most of us are like thinking, how or when will I even be asked this question? Is it even a possibility? Or maybe these researchers are just talking about their own things. But I can assure you that a decade from now, let's say maybe 10 years or less, this will be one of the questions that you will be asked in different contexts. For instance, you could be asked this question as a relative to a family member who has like a genetic disorder. You could also be asked this question as maybe your doctor suspects that you have a susceptibility to a certain disease and they just want to check. And you could be asked this question when maybe your hospital, your local hospital, wherever you are joining us from, Kenya, Nigeria, Zambia, Uganda, China, they could have already implemented these strategies, and now it is like a mandatory requirement in your routine checkup to also have your genome sequenced. So what is this thing that we are calling the genome? So I want to take us back. I'm sure that those in the molecular biology field, you're very familiar with this picture, which is showing there's a cell down there. For those who are not aware, if you can see my cursor, this is the cell, so it's an open cell. So when we go deep into the cell and we get into the chromosome, we get our DNA. So DNA is like deoxyribonucleic acid. It is the genetic material. It's like your genetic fingerprint. So as you can see from the title, the central blueprint of life. But those who are in molecular biology, we will you know that we refer to it as the central dogma. So the central blueprint of life, it means that this is where like we can be able to decipher your blueprint. You as Wangare, you as Effie, you as Austin. What is your blueprint? What makes Austin? What makes Wangare? And then this information is able to be translated into an mRNA, so that is just microribonucleic acid, and then we have this mRNA will later on be translated into a protein, so as you can see from the picture. So don't worry so much about this. Our panelists will put all these issues into perspective very soon. So the journey started from genes, which is just the unit of heredity. And then in the 50s, in the 90s, genetics was the buzz, where we would just sequence specific targeted regions of the DNA, and then we would discover traits, how traits are inherited, Mendelian theories and all those, which we are going to learn very soon, so just be patient. And then now in this generation, what we are mostly talking about is genomics, which is the study of the entire genome of an organism. So even that one, I just want you to have it at the back of your mind. Our panelists will explain in detail all these things and how we are even here and what do we mean when we talk about genomics. So how did we get here? So I just wanted to acknowledge a few technologies that have brought us to this point in time. So the first one is the polymerase chain reaction, which is available in most of our laboratories. And this technology enabled the ability, it gave us the ability to be able to identify a few targeted sequences and be able to decipher that information. And then later on, there were other technologies that were developed, like the Sanger sequencing, the manual one, which was very laborious. It had its issues, but it also contributed to some extent in some of the knowledge that we have now. And then came the automated Sanger sequencing platform, like in the next picture where you are seeing. And then this platform has also been deployed and it has also contributed to a lot of knowledge up to the extent where we are able to talk about genomics today. And then now to what we are really interested in, the greatest milestones in the medical field, especially in the genomics field, the next generation sequencer. It's such a buzz, this technology. And the picture here I have is from MySec. I'm not marketing them. It was just the most available picture to show what a next generation sequencer looks like. And then since we are going to answer this question, like you as an individual, being the person that we are sequencing today. So I'm going to be a bit biased towards the clinical perspective. And as you can see from the two pictures at the bottom, there's the HPLC. So this is an Agilent 1200 platform. So these two, the HPLC and the mass spectrophotometer, when genetic knowledge became available, what could be done at the time was just phenotyping. Like we would just be able to identify the phenotype of an individual or describe conditions at the phenotypic level. And these were the technologies that were available and are still in use in some institutions up to today. Then currently, in my field especially, we are using the Quant Studio 12K Flex. I'm not marketing them, but it is like the most practical example that I have because I have experience with the machine. And here, in this context, we are using it for genotyping. So like we've moved from your phenotype, which is what we can see, to your genotype. Like at the genetic level, what information can we get as is regarding your condition, your medical condition? So what brought us to this? Okay, 1 minute. Okay, so what really brought us to this step as Africa to even have this conversation on genomics? So we have like a few key projects that I really wanted to mention to you so that you can have them in mind. So the first one was the Human Genome Project, which was really a great milestone in the scientific world. So as technology was changing, as all these other fields they were developing, in the research, in the medical field, we were also developing alongside with the technology. And one of the greatest milestones, especially under in the clinical circumstances, is the Human Genome Project, which sequenced the first blueprint of a human being was sequenced through this Human Genome Project. And then after the Human Genome Project was sequenced, it gave birth to other projects that are of clinical relevance today. And the first one was the 1000 genomes, just to mention a few, the 1000 genomes project, which tried to fill in the gaps in terms of variants. And then we have the H3A Africa, Human Heredity and Health in Africa, which I'm part of the beneficiary. So this project was instigated to be able to create a collaborative effort between African researchers. So it's basically being funded by NIH and Wellcome Trust, but it is the African researchers that are running most of this research that is ongoing under the H3 Africa umbrella. And then we have other projects like the Genome Ad, which is a database that contains all this information. Another notable project is the Malaria Gen, Genomic Epidemiology Network. So the Malaria Gen, it factors diseases such as malaria, HIV, tuberculosis. These are like infectious diseases which are unique to Africans. So as much as it is also being funded by NIH and that is National Institute of Health and the Wellcome Trust, most of the research work is being done in Africa by African researchers. Then the next one is the TopMed. So this is transomics in precision medicine. So precision medicine is also, as you have seen from the flyer, we have a precision medicine advocate as one of our panelists and a precision medicine champion. So they will tell us what is it that they mean by this precision medicine. So stay tuned. And also one of the new baby in town is the DSI Africa, which is seeking to build capacity in terms of data science for African researchers. And then we also have the UK Biobank, which is the second last one. And this one, it is like a very huge database that has a lot of information, clinically relevant information. So if you're a research lab or a research sector that you're doing your own research in human and you want to get a database where you can compare your data sets to identify a few things, then you would talk to the UK Biobank. And of course, the last one, which is the most talked about, especially in my field, that is the 3 Million African Genome Project. So these whole projects, the way they've been run, the infrastructure, the framework, the setup, they have given us a reason to want to do this project, which is a project that one of the core aims is to create an African reference database. And Professor Ambrose Wonkman from South Africa is really passionate about this, and he talks about it. So if you really need literature to go around and see what it is all about, I can always send the reference materials. So the next thing is the application of genomics. So the first field, again, I'm being biased at this point, I apologize. So it's personalized medicine, and we will learn about all about it in just a few minutes, so just bear with me. And in personalized medicine, we can be able to look at genetic disease risk. We can be able to tell you, you see, you as, unfortunately, I'm just seeing Austin and Effie. We tell you, you as Effie, you're at risk of developing diabetes, or you as Austin, you're at risk of developing this disease. So this is personalized medicine for you. Then we also have ancestry. If you want to know, we have companies like 23andMe, which can tell you like maybe you are 30% African, 20% European, 10% Indian. So if you want to find that information, this is personalized medicine for you. Then there's genetic fingerprinting, whereby you can be able to trace, maybe at a crime scene, we can be able to trace whose DNA was found there, something like that. And then most of us as young, I think people my age, you are willing to get, it is your hope to get married at some point. So you also need to check whether you're genetically compatible to your partner. So now we're even bringing genetics to courtship, because it's a very important question, as we will see later on. And then paternity testing, which is also a buzz, especially in the African context, is also part of personalized medicine. Then the picture on my left, if you can see my cursor, so this is just a roadmap of how we are able to track COVID-19 variants, which is one of the most spoken about diseases at the moment. So the knowledge in genomics can enable you to track the variants. So going outside the clinical perspective, we have down here, you can see, it looks like that structure looks familiar, right? Maybe tell me in the chat box, what do you think that is? Have you seen that structure somewhere? Except in this context, it is made out of fruits. So that is just the normal helical structure of DNA, and we have a new field that is now called nutrigenomics, whereby we are able to prescribe what sort of foods you should be eating as an individual based on your genetic information. Then the next one is at this corner, those people who are working with plants, you can be able to tell us that you can use genomic knowledge to identify certain plants that are varieties that are maybe extinct, or to find those ones that are medicinally useful. I'm not an expert in that field, but I'm sure if any of you in this meeting is a plant expert and you're using genomics in your field, you can comment on the chat and let us know how are you using genomic knowledge. And then down there we have two people shaking hands, so two hands that are shaking, and here we are showing how you can use genomics knowledge in the microbiology experts. You can actually use genomic knowledge to try and trace transmissibility and see, maybe if you're in a hospital setup and you wanted maybe there's a staphylococcus infection and you want to trace where could the infection be coming from. Is it the lab? Is it the reception? Is it the maternity wing? So you can be able to do this. And then I'm sure somebody is wondering, why am I seeing the helical structure in a cow? So this one is actually a very interesting story. So some students in our lab at the African Institute of Biomedical Science and Technology, high school level, they were able to sequence the DNA of a cow. And then they were able to also convince us that using this information, if your cow is stolen today, you can be able to go and trace it, and then you say, this is my cow. And then if they ask, what evidence do you have to say that this is actually your cow? You can provide the genome sequence and say, according to this information, this is my cow indeed, and you can do that case in court. So I don't know, maybe it's something to think about. So genomics is way, way closer to you than you think, and it is not something that is way too new. You are just not aware of it yet. So now that you have had a bit of a feel of what is genomics is, so I would like us to take this opportunity to participate on the next poll. So just remember, you have to log into Slido and then join the quiz. I want to see a few people who have joined the quiz before we proceed, because I want to have your thoughts before I invite the panelists, so we can see if maybe they will convince you to change your mind, or you would just still be willing to stand with your sandwich with the current
Maybe
knowledge
you
that you
can
have.
reshare the link, because we have some new people who've just joined us.
All right, all right. So I've just reshared the link. I'm seeing Wangari is there, PK. Interesting name. Isn't that a chewing gum in Kenya? My Kenyan people. Lawrence is in. I'm seeing Winnie, a few more people. Ahmed, thank you for joining in. Auri, thank you. James, thank you for joining. A few more people. Yes, Godfrey, Thomas, Vinakesh. Okay, so I'm going to start the poll. So don't worry, the polls are anonymous. We won't know you're the one who voted. So in case anybody is having second thoughts on it, you can just go ahead and join. So do you want your genome to be sequenced? The question can be framed in different ways. So do you want, now that you know, you can even use it to trace a cow if it gets lost, what about your own information at this point before I welcome our panelists? Okay, let's see. A few more votes.
So I noticed that some people are typing in the chat. Maybe
All
they're
right.
having challenges getting
Joining.
onto the link. So if you're using a phone, if you're able to minimize your Zoom app, then you can go on to your normal browser. I don't know what you use, Mozilla or Google Chrome, and then type in, copy paste this from the Zoom chat onto a different tab on your computer, on your phone. But if you're on a computer, it's easier. It's actually easier. You just minimize Zoom, open your Google, just your normal Google homepage, and basically copy paste, and that you'll be inside the slider. So you cannot type into the Zoom screen. I think that's what people are confusing. So,
Oh, sorry, guys. So
yeah.
whatever we are seeing on the Zoom screen is from information that people have voted in the Slido. So we can see no is standing at 11%, and yes is standing at, let me see. So maybe the people who are saying yes, you can send a few comments in the chat to tell us maybe in one word or two, or maybe in a sentence, why are you saying yes? And those who are saying no also, I would like to hear your view on the issue as well before we invite the panelists. Okay, so just keep voting. We will return to the poll and see what the rate is at the end of the presentation. So this will take us to the next session, which is like the most awaited for session, whereby I'm going to invite our panelists. So first panelist, Miss Glory, you can switch on your camera perhaps. So our first panelist is Miss Glory Jayeoba. So she is currently carrying a BSc degree in zoology from the National Center. She was an intern at the National Center for Genetic Resource and Biotechnology, and she was also a lab technologist at the Federal University of Lafayette in Nigeria, and she's currently doing her master's degree in genomics and precision medicine at AIBS. So welcome, Miss Glory. Maybe you can wave at us so we can see. Okay, thank you. And then the next panelist for this session is Miss Neema Hadara, who holds a bachelor's degree in biological sciences. She has had experience at the medical research through an internship at the Kenya Medical Research Institute in Nairobi, and also at the University of Nairobi. So University of Nairobi people, hi. She's also currently doing her Master's of Science in Genomics and Precision Medicine at the African Institute of Biomedical Science and Technology on an EDCTP scholarship. So welcome our panelists. So I would also like to remind us that in the Slido link, for those who are still on the web browser, you can send your Q&A, your questions there. I will be able to read them. And you can also send your questions directly to the chat, because I will be asking those questions in due time. And we also have a Q&A session at the end of the discussion. So feel free to ask a question at any point in the discussion session. So my first question is, what is this genomics, Ms. Jayeoba and Ms. Neema? What is this genomics? And why would I, as Rissy Makokha, want to have my genome sequenced? Like, why would I want that? Like, I'm a healthy individual, I'm doing well, I'm doing fine. Why would I need to find out this information? Or do I have to, unlike the cow, I know I cannot be stolen. Can it be used to trace me? I don't know. You guys will let us know. So I don't know who's ready to give it a go. Let me see. Okay. So I will give the opportunity to Ms. Jayeoba to give us his views on the issue on what is genomics and why it is important, and why I, as Rissy, should get my genome sequenced.
All right. Hi, good morning, everybody. So, like Ms. Makokha said, my name is Jayeoba Glory, and I'm from Nigeria, but I'm currently doing my master's degree in precision genomics and precision medicine in Zimbabwe. And first of all, I would like to apologize for coming late. I'd actually tried my link ahead of time, but my laptop decided to show me who was the boss this morning, so but I was able to overcome it. So thank you so much for waiting. So down to the business of today, we're talking about genomics, and I'm sure we're all curious about what this genomics is. So I'll just start by defining what genomics is. So according to the US National Human Genome Research Institute, that's in the US, right? They define genomics as the study of a person's, like all of a person's genes, and also the interaction between these genes. So when we're talking of all of a person's genes, we're talking about the genome. So if I would define the genome, you say genome, like someone put in the chatbot box that they're curious to know what a genome is. So a genome is a person's complete DNA, and it contains all of an organism's genetic information. And this is very important we discuss about it, because it is an important determinant of our health. So, and I'm sure at a point also in time, we might have come across this omics, like apart from genomics, we've heard of maybe proteomics, metabolomics, and even microbiomics. So all these omics, like what exactly are they? So in life sciences, or in biology particularly, we use the term omics to describe any field that deals with a totality of something big, something complete. So we have like our genes, right? So when we start considering all our genes as human beings, then we're starting to talk about the genome. Then similarly, when we start talking about genetics, which is the study of our genes, but when we start considering studying all our genome, then we talk about genomics. So we have genomics, genetics, genomics, and these terms they're like say intertwined. So we can't really talk about genomics without talking about genetics. So genetics is like the foundation for genomics. So now the question is, when did genomics really start? Because it sounds like it's something new that just started recently. So it's partly true, because it's a field that is emerging, but like I said, since genetics is the foundation for genomics, so it means to talk about genomics we have to talk about genetics and how it started. So mind me as I take you down back to history, down to 1865, when the father of genetics, who is Gregor Mendel, discovered like the principles of inheritance. So why is Mendel important in this story? before Mendel and his discovery, most people believed in like the theory of blending inheritance. So what I mean by that is that, so they say when maybe a child, let's say the father is very tall and the mother is not so tall, so they believe like the child's height will be somewhere in between the father and the mother. So that's like the blending of the height of the father and the mother. And it's really interesting to see that people actually thought like that before. So but Mendel, when during his time, he actually was able to prove with his theory a genetical basis of inheritance. And like the way his theory was stated, he said there is a unit factor that is responsible for heredity. So he didn't know what this was, he didn't know whatever was responsible, but he was able to show that yes, there was there was something responsible for heredity. And this was further like proven by Thomas Morgan when he came up with the chromosomal theory of inheritance. So all this like brought about curiosity in the science world, and they went out to keep searching of what exactly is this unit factor. And that was when DNA was discovered as the factor responsible for heredity. And if I would summarize the whole story about the discovery of DNA, I think I would have to mention Cosell, who discovered the 5 nucleobases that constitute our DNA. I think we know it to be like adenine, guanine, cytosine, thymine, and uracil. Then we have to talk about Edwin Chagaf, who actually discovered the base pairing. So he was able to say that, well, adenine should bind with thymine and guanine should bind with cytosine. Then we have to talk about Avery and Griffiths and Master Chase and Hersey, that all together came up to discover that DNA is the unit factor responsible for heredity. So, it started with Griffith's experiment. So, in his experiment, he came up with like a transforming principle that there was something that was able to transform the non-virulent pneumonia strain to a virulent pneumonia strain. So, Avery took this research further by discovering that it was DNA and not RNA or protein that was actually responsible for this transformation. Then it was finally confirmed also by Hersey and Matachase when they used radio labeling to actually confirm Avery's experiment, excuse me. Then I think we have to also mention Francis Crick and James Watson, who were the ones that came up with the structure of our DNA. So after this era of DNA discovery, then we have the era of when the technologies, the molecular techniques came about, like the restriction enzyme that now we can also use for genotyping. Then we have the DNA sequencing technique that was discovered by Frederick Sanger. Then we have, Frederick Sanger came up with the manual one with test tubes, with the gel slab and all these forms. But it was taken further by Laurie Hood, who came up with the automated version. So what we see now in our laboratories, the big nice instrument, also it started by Laurie Hood. Then we have to talk about the most popular, the PCR technique, that I'm sure like almost all lab nowadays should have a PCR machine, and it was discovered by Carrie Mullis. So all these amazing theories, so like Miss Makokha had already mentioned, so with the advancement in technology, they were able to, especially with the DNA sequencer, they started sequencing just genes individually like that. But at a point, I'm sure the scientists came up with this crazy idea. They sat down together to think that, well, why don't we try sequencing the whole genome, like all the genes in human beings? So that was where the Human Genome Project started. So it was a crazy project, I can assure you, because at that time it did not look like possible, it did not look possible, but it took them 13 years and it cost them billions of dollars, but it is really, really much appreciated because this single project changed the course of history. So it was at this point we started shifting from genetics, just the singular genes, to genomics. So we can say the shift when that was it happened when they decided to sequence all our genomes. So we call this the genomic era, like the beginning of our genomic era. So the purpose of us going down history is not just to bore us because I'm sure I know history is a bit boring, but for all of us to get the whole picture of how we got here as in science, in genomics. You know, when we talk about these machines, we just open them and close them, and in 3.5 hours with our PCR machine, we have multiple copies of DNA, and it looks so amazing, but we don't really understand how we got there. So us seeing this gradual process of in genomics is just showing us how far we've gone. And I think we really need to applaud the science world because we've made serious progress in genomics. And also, I just want to also mention that going through history and studying scientific discoveries in general, it helps us to understand that most scientific breakthrough is more of a cumulative effort, like cumulative contribution of individual effort. So it's not just one person standing with the trophy calling himself the god of science, but it's people, many people coming together, rubbing their ideas to come up with a solution to a particular problem. So like we can talk about the DNA. One person came up with the basis, another person came up with the base pairing, another person came up with the structure, and all in all, we know it's formed what we know as DNA today. So it's basically everybody's individual work adds up to a body of knowledge until a research question is answered. So I'm mentioning this so that we understand. I'm sure most some of us are in the science field, so we know our contributions to science, that little project we are doing, that little research we are doing is really important because what you find might be the key to what another person will need to come up with a very big discovery. So fast forward to today, where we have the amazing technologies. So it's just to tell us that where we are coming from, where we are today, and let's look at the future. How do we see the future to be in genomics? Like now we have so many big, before you can sequence your genes in for days, but now you can get it in less than 48 hours, which is amazing. So now, so looking at the future, I'm posing this question to us now. How do we see the future in genomics, and do we see ourselves to be partakers, to be major key players, to contribute also to the body of knowledge in this field? Thank you.
So thank you so much, Miss Glory, for that detailed explanation and taking us back history lane as to how we started with the gene, went all the way through genetics, and then now we're even talking about genomics. So I can just read you one comment on the Q&A section. It's from Wangi, and it's saying history is not boring. When I teach maths, I have to go to it. When I teach economics, I have to refer to it. So when I teach biology, so genomics history is fine. So I think you're safe at this point. And I would also like to read a few interesting comments on the chat before I also summarize on what you've just told us. Somebody is saying they want to know their genomics, have their genome sequence so they can see if they can eat more chicken. I don't know if that applies to you. Miss Marie, maybe you will explain to us later on how that is very relevant to you. And I'm also seeing some other great points in the chat. Um, Such as some people are really glad that genomics is actually being done in Africa. Somebody says they want to know their risk for prostate cancer because it's now ethnically related as an African man. That is interesting. And Dr. Doris Njoka is telling us she would like to know whether learning about foods that are fit for her so she can stop working out, I'm guessing. That is really interesting. So thank you so much, Miss Jayeoba. As we have just seen from your in-depth discussion, you have even told us of how different individuals have actually built on this knowledge and made sure that it is not like a one-person effort, but multiple individuals contributing to this field of knowledge bit by bit until we are where we are right now. So I hope that at this point you have been able to actually appreciate like the milestones that you have taken to be able to get us at this point. So those individuals who are not yet sure whether they want to have their genome sequenced, I hope that the additional information that you're going to get from Ms. Neema Hadara would be able to convince us. And if you're not convinced, please just keep staying in and convince you that you should have your genome sequenced. Am I convinced yet? I would like to reserve that information to myself for now, but you can let me know in
Is it just me or is Rissy's video buffering? Rissy, I think your video is buffering. Can everybody hear Rissy?
No.
Okay, so
Yeah.
it's not my internet. Okay, all right. I think we lost her connection, but she'll be right back. No problem. This is an interesting discussion. I've always used the blended theory. I also always just thought, a child has to be medium if the one parent is tall and the other one is short. So if you're like me, you're not inside of this special field of knowledge of genomics, don't worry, you are at home. This session is both for the specialists and the generalists. So we are here to learn. No question is bad. No question is too crazy. So feel free to ask anything at all. I really like the overview that the team have given us so far. Very good job. And Glory coming in from Zimbabwe, meaning her time is actually an hour earlier. That must be really early where she is, and giving us such a nice history so far. So what are you getting? What are you learning so far? What questions have you always had? I definitely have had questions about this whole GMO stuff, about the food we eat. We go to the market, you're not even sure anymore what you're eating and how it's related to your risk for cancer. And so I think I also want to be empowered as a consumer of food, as to what to do in those instances. So I'm just checking to see if Rissy is back. Rissy, let me know when you're back. I see Neema is still here, but Glory, I don't see Glory. So Glory, are you here? Yes, I think it's technical hitch. So tell me, what is your interest in this whole specialization? Let's make this interactive. Feel free to unmute. We are not so many, or you can type in the chat. Ahmed Abi says it allows us to examine certain specific research studies like virus or species. I like that. Yeah, so it's quite precise field of study. I think the others had been written. Nyangwara's is funny. He wants to know if he can eat more chicken in peace. Nowadays, we know that the food we are eating, and there's a food revolution now going into looking at the genes and what's going on there. So who else would like to tell us who's gonna go next? Is it Sally Rono? Is it James? Who else hasn't spoken? Afolabi, tell us. Akech, welcome. Had you unmuted yourself? I think you are unmuted. You can proceed. Yeah. Yes, welcome.
Okay, thank you very much. It's a very interesting session we are having, especially on the part where the cow was stolen and the students were able to trace their cow.
Oh,
that seems interesting.
That is quite precise. It's a precise science. You can trace the exact cow that was stolen. And for most of us in the human species, we think cows look alike, but DNA will tell you which cow was stolen. So
yeah. I never knew that was so much applicable.
Right.
so interesting and I enjoy it every moment. I can't wait for them to continue.
Right. Yes, Glory, I see you're back. I'm not sure if our moderator is back, Rissy. So I'm just
Maybe
waiting.
Neema, you already knew what to talk about. Maybe
Oh,
Neema
good.
could continue.
Sure, Neema, welcome.
Okay, thank you. And good morning, everyone from this side. As you've been told, I'm Miss Neema Hadara, and I'm currently pursuing my master's in genomics and precision medicine here at African Institute of Biomedical Science and Technology. I don't know if I'm clear.
Yes, we can hear you, we can see you. Yes.
Oh, okay, thank you. Yeah, so thank you to Miss Rissy, okay, she's still out. And we are sorry, by the way, for the technical issues. That is technology for you. sorry about that. Yeah, so thank you, Miss Rissy, for the wonderful introduction you've given us, and Miss Glory Jayeoba here for the wonderful walk down the history history lane, just to tell us where all these genomics came from and what genomics is all about. Yeah, history too has never been my thing, so yeah. Yeah, so thank you for that, Miss Glory Jayeoba. Yeah, and maybe just to recap a bit from her great definition there, that genomics is simply a combination of the word genome and the omics, where the omics part means a totality of something, a totality of some sort, and genome means all of the organism's genes. So it's basically the study of all of the organism's genes in total. Okay, so I think I'll basically be concentrating on the molecular aspect of genomics. So just to add on what Glory has talked about, I will simply start by emphasizing that maybe we should not confuse genomics and genetics in the first place, because genetics is simply genetics simply aims at studying the individual genes and how they are inherited, while genomics here is focusing on studying all of the organism's genes, how they interact and how they are expressed. Yeah. So basically a gene here, because I know in our participants here, this audience here, in our audience here, I'm sure not all of us are biologists, okay? So yeah, I'll be taking you step by step to really understand what we are really doing in genomics. So basically here, a gene simply means it's the unit of inheritance of a living organism, and it's found in the DNA. I think my colleagues captured what maybe DNA is, what DNA means earlier on. DNA, that is the deoxyribonucleic acid. So the gene simply is the unit of inheritance, and it is found in the DNA, which is embedded in the cell nucleus in the form of chromosomes. And basically, it's usually estimated that we as humans, I'll be concentrating more on the human side. So it's estimated that we as humans, we contain around 20,000 to 25,000 genes in total. Okay, so you can imagine now in genomics, you are studying all these genes, the 20,000 to 25,000 genes in the human body. So DNA is a double helical structure, and it contains a sequence of nucleotides. And by nucleotides here, I mean base pairs, okay? The base pairs are adenine, guanine, thymine, and cytosine, okay? Those in molecular biology, I know this is your area, you know? Yeah, I know this is your area. So yeah, adenine, guanine, thymine, and cytosine, which are usually represented in simple letters A, G, C, and T. And they always have a specific way of pairing according to Sir Erin Chagaf's rules, as Glory captured it in the history bit, that there was a great person called Erin Chagaf who discovered the base pairing. Okay, he discovered how these bases are with each other. So the DNA is usually a double helical structure. So yeah, these bases are on either parallel strands and they pair with each other. So usually A pairs with a T and a G with a C. So therefore, I hope we are now getting a picture of a picture of the DNA. So it's a double helical structure and it has the base pairs, the nucleotides of the base pairs pairing with each other. So therefore, a specific sequence or arrangement of these nucleotides on the DNA represents now a gene, okay? Yeah, a specific arrangement of these nucleotides on the DNA represents a gene. Then now, because the DNA is very long, so that is where we find there are several genes on the DNA across the DNA, which again they have expression and transmission of specific traits of an organism. Okay, so that is the gene for you and the DNA. So therefore, for us to study the genome, we basically need DNA, okay? We've said the DNA is where the genes are contained, and we are studying basically all the genes of the organism. That is called genomics. So we basically need DNA. So DNA here can be extracted from any body tissue, organ, or blood of the organism, since the DNA in all the cells of an organism has the same sequence, okay? It has the same sequence, and by sequence here I mean the arrangement of those nucleotides, as I've said earlier on. Yeah, so studying the genome here basically means studying this DNA to see how these specific nucleotides are arranged, and this is now what we call sequencing. Okay, so sequencing, we are studying how the DNA nucleotides are arranged, and I said they're represented basically in simple letters A, G, T, and C. So we are just studying this arrangement of these letters. That is sequencing. So it is also important to note that modern throughput sequencing methods, generating sequencing, and maybe NOPO and many others are faster and cheaper. Okay, I think Ms. Rissy captured those technologies earlier on. So the modern day sequencing methods are throughput, and they are faster and cheaper. So hence you can have your genome sequenced in just a few hours at an affordable price, maybe less than 1,000 USD, right? So yeah, you can have your genome sequenced at a faster rate and at an affordable rate. So it is now in these sequencing results, after we have sequenced your genome, now we have your results. So it is in these sequencing results now that this is where now genomics starts to make sense. So with the sequencing results, this is where you are able to obtain useful information, such as maybe now specific variations, okay? Specific variations in the sequence. We are able to identify specific variations and their functional effect. Okay, so we do this by comparing sequences, mainly with what we call a reference genome. Okay, so usually, like the human genome, the first genome that was sequenced, so that is our reference genome. So any other genome that we sequence, we compare it to that one. So that is the reference genome. So we compare it so that we can identify variations, okay, in our sequences. So after comparing, maybe, as I've said, that is where we can identify variations. So maybe at a particular position in our reference genome, maybe C was pairing with G in this particular position, but now in the genome we've sequenced now, it is an A pairing to a T. So that means there is a variation at that particular position. Okay, so then the functional effect of these variations now can give an explanation now to our differences, okay? Yeah, e.g. maybe as humans, our differences in maybe hair color, eye color, skin color, and so on. Yeah, so those variations now are explained, the functional effect of these variations is explained through that. So what I mean here, we are able, for example, we are able to identify that maybe the hair color of a particular people is because of a variation at a specific position of the hair color gene and etc. Right? Yeah, so we are able to identify variations now through sequencing. So it is also confirmed that as humans, we are usually 99% similar, okay? 99% similar. So that means there is we are 1% different. We are 99% similar and there is 1% different. And this 1% now, it accounts for our differences, okay, as humans. Yeah, as I've said, differences is such as the hair color and all these things, skin color and all these things. So it is accounted in that 1%. So, and this 1% is usually equivalent to around 3 million nucleotides or base pairs. Those A to C, the letters I've just talked about, it's equivalent to 3 million of them. So what I mean here by 3 million base pairs is we have a total of 3 billion base pairs. Okay, we have a total of 3 billion base pairs. Those letters in the DNA, there are 3 billion of them. So that means the DNA is very long. Okay, it's very long, but it's usually compacted and found in the form of chromosomes, so that it can fit in the cells. Okay, so we usually have 3 billion base pairs, and now that we have two chromosomes, it totals to 6 billion base pairs. So now that's why that ends why Sync produces amounts of data, because you can imagine analyzing 3 billion base pairs from a certain genome, and here I said I'm basing on the human, on the human genome. Okay. So sorry. Yeah, so that is basically all about sequencing and all about genomics. Yeah, so as I've said, we're basically studying the arrangement of our nucleotides and to see variations. And through studying this, now we are able to studying the variations, we are able to see if somebody now is maybe susceptible to a certain disease, what causes a particular disease disorder, and all that. So there are a lot of things involved. There are a lot of things there's a lot of useful information we can gain from identifying these variations. Okay, so there are many things, yeah, we can get from that. So I think that is it from me, and thank you. I hope you've gained something.
Thank you so much, Ms. Neema, for that detailed explanation on what genomics is all about. So I hope you've all been able to at least gain one information or two from that brief description. I'm not sure what brief in what context, but I hope you've gathered something. So I'd just like to read a few of the comments. So Ms. Sally Rono says, this is very interesting. I'm a finance person, and maybe you can just explain the financial application of genomics to the economy of the nation. Who would like to take on this, Ms. Neema or Ms. Glory? Kindly unmute yourself, Ms. Glory. Okay, as Ms. Glory is dealing with the technical issues, Ms. Neema, kindly take on this question.
So I think genomics is important in every aspect. So you've chosen the economic aspect, okay? So I think for the economic aspect, it can be important, like maybe through creation of maybe jobs, because genomics is an interdisciplinary, okay? So it doesn't just involve maybe as lab people, okay? It involves so many people working around it. It will require a statistician to analyze data. It will require the lab scientist as well. It will require so many people. So through this, I think we are creating jobs even for our people, and yeah, they can benefit economically. Yeah, and yeah, it will help to boost the economy of the country. Yeah, I think I can continue.
I totally agree with you on that one. So
So
maybe
maybe
just
just
to
to
add,
add,
the
the
financial
financial
applications
applications
of
of
genomic
genomic
comes in the
comes in the
context
context
whereby
whereby
we need funding for us to be able to run these projects, and that funding is going to come from you people, and that way the financial analysts, they also need to sit with us and help us. When we want to create infrastructure, we need funding to do that. So those are a few of the applications of genomics, financial applications of genomics, because it's actually a financial burden, and most of these processes are very costly. So they do not come in cheap, but just stick around to find out how much would it cost. But you're also trying to make sure that the cost goes down over time. That is what we are aiming for. And another a comment, maybe if I may read, just a moment. Mr. Page next, Chikondoa is saying, quite intuitive. This is really interesting stuff. So now it means genomics is not something new. We are glad to bring this knowledge to you, Mr. Page next. You're very welcome. And let me see another one. So you can keep sending your questions in the chat and your comments as the session is proceeding. Uh, so Ms. Wangari is saying, so genomic sequencing of my cells would only reveal the predisposition or risk at a percentage probability for, say, genetic disease for my family tree, but not absolute possibility, right? I don't know, would somebody else like to comment on this? Um, all right, so I think I'll take this one. So I believe that that is actually true, Ms. Wangari. As much as from what Ms. Neema has just described, as much as we are 99% similar, it is that 1% that causes this variability, and this variability is like for you as an individual, you are way different in terms of a huge percentage as compared to the next individual. And this small variability, this small percentage is what causes that probability of you being able to maybe develop a risk for a certain disease, but it is not certain. Just remember that this is research knowledge, but we need to back this up with clinical information for us to be actually say like, yes, you do have this risk, and this risk actually has an implication on your life. So how much is paternity DNA test in Kenya? Kenyan people, my Kenyan people, would anybody like to respond to that one? I'm not really sure. I'm not sure, so I will not comment on that one. Somebody also says it can help prevent diseases because one can intervene early. Curative care is more expensive. That is true. It's all about prevention care, especially for diseases like the rare diseases like diabetes. If you already know you have like risk for diabetes, maybe you might start doing lifestyle changes like working out constantly, taking the right food to avoid being obese and all, so that you can help. So you don't need to wait until you are sick for you to like get treated. You just have to start early. Then maybe let me see one more or two more. If I've skipped yours, I'm so sorry. Uh, if I've skipped yours, um, let's just see if I can get a few more. So Lawrence is saying, very interesting to know that this 20,000 to 25,000 gene is on the chromosome in the nucleus, and we get to know and see the sequence of what we can't actually see without all these technological tools. That's actually absolutely true. I totally agree with you. It's very interesting indeed. Let me see another one. Wangari also says, in my understanding, science can describe the patterns and functional effects of differences, but may or may not be always able to explain the why. Is genomics able to only describe the human genome in genetic diseases, but not explain why the person was born that way? Is that correct? So from my personal understanding and not the view of the field is that, yes, genomics can be able to explain a few of these things. Like, as we just said, this field is very new and is when we are trying to gain more information. And for us to gain this more information, we need to have more data. And where is that data going to come from? It's going to come from you people agreeing to go through sequencing for your genome to be sequenced so that we add into the data. So as time goes by, we will be able to understand the genome more, but the little that we know so far, what we are able to do with that knowledge is actually assuring us that we will be able to even achieve more as more understanding is gained. But that is actually true. Most of the things that genomics information can give us, we cannot actually be able to explain them. Another thing, you mean differences in human is just 1%? Yes, that's true. So that difference is what is really causing us to be different. That is why some of us are light skinned, some are dark, some are tall, some are short. Even the differences in ethnicity can be described by that 1%. I'm seeing an ethical issue, which is great, but just hold on. We will see how it works. Let me see another one. So Mr. Austin is saying there are several cases of violence in households as a result of failure to ascertain the paternity custody of many children. Hence, the study on genomics ascertains through DNA tests to reduce such cases. The problem is that the knowledge about this is still limited. Well, yes, that is definitely true. I totally agree with you, Mr. Austin. This paternity issue, especially in Kenya, the rate of paternity fraud has really gone up, and genetics is really helping to give us information on these tests. And also a lot of family issues are around it, and we cannot just we do acknowledge that genomic knowledge, it has some benefits, as we are going to see in the next few questions that are coming up based on your questions, but we are also able to tell that as much as we are trying to solve some problems, there are still some solutions that we don't really provide, like the implications of this knowledge on other individuals, on the families. Like, you will come, you say, okay, I want to know if this is my son or this is my daughter, and we will give you that information. But we ideally need to work with genomic scientists, like with the genetic counselors, who will be able to understand how you reason as a person, how will you handle the information, and be able to work with you through that process. So I'm seeing Miss Glory is back with us now, which is good. So Mr. Hamed is saying, can we identify virus variants by using genome sequencing like COVID-19? Ms. Glory, would you like to take that one? We've missed you. Can we identify virus variants by using genome sequencing like COVID-19?
Okay, yes, we can. So it's basically that is what has been going on throughout this pandemic. So most of the times this virus is sequenced, and based on the analysis, because I think we need to mention that sequencing our genome is not just the end game. There's nothing much you can just get from just staring at billions of thousands of letters, so they need to be analyzed. So based on this analysis, there are a lot of analysis we can do with these letters. So one of them is called the phylogenetic analysis, where we can do the ancestry trace. So that's how they can trace that, oh, this variant is from China, this one came from India, this one came from New Zealand, and what we've been hearing so far. And also we can also use this genome sequence to identify the various strains. So, based on the mutation, we can see. So that's how we'll be able to know that, oh, this one has mutated because there's a difference in the base pairing here in a particular position. I know, and as we talk later, we'll talk more about the implications of variation. So that's how you will hear that this strain is more virulent than this one. It's based on the analysis in the genome, and we saw some variants that, should I say, gave this virus a gained ability that made them more virulent. And I think I can mention that even with sequencing of our genome, it helps in vaccines and drug discovery, although I'm not really an expert in that field, but I can just mention that with our genome, we can actually identify target sites and how we can use it using this bioinformatics. So I'm glad I saw some statisticians. So there's fields in with the computer science field, the bioanalysis, biostatistics field, where they use this computational biology to actually, like, should I say, simulate how this virus will interact in the body that can help them with drug discovery. So that I think there are a lot of things we can use the genome, and this is just a virus. So think of what we can use our human genome to for also.
So thank you so much, Miss Glory, for that one. Yes, indeed, as you've just explained, you can be able to detect these variants. Where actually, we can give like practical case examples, like South Africa and Nigeria were like the first to among the countries to be able to sequence this variant just when the disease started. So that means that this time Africa was not left behind. We were actually part of the process, and we enabled these variants to be sequenced, and we were able to trace them. So that is why we even need more individuals to join the field, so that next time, in case such an occurrence happens, we can be able to see a variant is sequenced in Kenya. Kenya is releasing sequencing data. Uganda is releasing sequencing data. Zimbabwe is releasing sequencing data. Zambia is releasing sequencing data. So that is really important. And then there's an interesting question that just came in. I think maybe I just saw it now. Is it ethical not to marry someone because they have a genetic disease predisposition? Is it ethical not to marry someone because they have a genetic predisposition? And is it ethical to abort a baby in utero because they have been found to have, let's say, Down syndrome? So these two are actually very, very interesting sessions. So as I give our panelists a chance to answer the questions, I would like to also know a few of your comments on the comment section. So is it ethical not to marry someone because they have a genetic predisposition? Who would like to take this one first? Ms. Neema, would you like to give us your comment on this?
Oh, okay, sorry. Yeah, I think that's a tough one. Okay, because in ethics, everybody has a right. So, uh, what was the question again? Is it ethical to marry someone? Okay, so you're asking if it is ethical to marry somebody if you discover they have a genetic disorder. Okay,
To
so what
not
I'm saying
marry
here,
them. Sorry, the question
or
was,
not.
yeah, to choose not to marry them if you go do a test and then find, oops, this family, they have this and that and a rare disease or something, so I decide not to marry them. What's the ethics there?
Okay, okay. Okay, so I think genomics can really say that, yeah, you should not marry them because you risk passing on these variants to your generations. Okay, but now otherwise in ethics, as we will see later, because you're going to cover that topic on ethics, we'll see later that everybody, it's like they have a right. So you have a right to choose whether you will marry them or not. But the genetics here, according to genetics here, the genetics here will just tell you that, yeah, you will risk passing these variants to your offspring. So it's up to you now to decide. So I think that's basically what I can say. It's up to you to decide, but from the genetic side, we will tell you that you will pass these variants to your generations and generations. You'll keep on passing these variants.
Thank you for that
Yeah,
question. And I
so
think
you
it's
have the
also
right to
interesting.
choose, the right
Thank
to
you.
autonomy.
I think it's an interesting question because then should we test? Should we be testing people if the goal of testing is to abort the baby with Down syndrome? I think that's also related to that question.
Okay. Yeah, definitely it is indeed interesting, Wangari. That's why I was actually mentioning earlier on that as much as we give this genomics knowledge, we still need to work with other experts in the field to be able to decide, like, is this information ethical? Because for us, our job will just be to tell you have the variant, or it is predisposing you to this, or your baby is going to be born with this syndrome. But the consequences of that, unfortunately, it is too much for us to handle at the moment. We cannot be able to handle that, but I would like to hear Miss Glory's input on this one as well.
All right. Ethics. I would just say ethics is it's a very complex field. Like, so if it's really, really complex, honestly, because when we talk about ethics, we're talking about the rights of individual, and in this case, everybody have their own rights. And like there's this word they use in ethics about this, what is morally right. So what you think is right might be differ from what another person thinks is right. So the complexities of ethics, so it's just like bringing a balance into, so there's something we call the benefit and non-maleficent and the beneficent. So it's bringing a balance of doing good and not harming. So I think in this case, generally it's more of the balance, and I feel that in case of the abortion or the marrying someone that is not that has a particular variant, at this point I think it depends on the person's choice. So I want all of us to understand that we all have our own choices. Me, I have my choice. So if I choose to marry someone that has a variant, it's my choice. If I choose not to marry anyone because of this variant, I think it's also my choice. But generally it's just my own choice and it's just my own view. So but that's like Rissy said, we in genomics is an interdisciplinary field where we have to work with everybody, in the psychologists, the experts in this field, so that they are the ones that will sit down to think of what is actually what the like to come up with, should I say, a common ground in this case. And I feel that is just basically ethics, finding a common ground to solve all these problems.
So yes, Miss Glory, I indeed agree with you, because here we are now seeing the issue of, you see, the child also has the right to be born at the end of the day, and you as the parent are feeling like, am I capable to take care of this child? And you decide, oh no, I cannot do this. So like there are a lot of ethical issues that are being raised because of this genomics test that we are doing. And especially on that one, I would also like to add a comment on the first one, where you should not marry someone because like their genetics say they're predisposed to a certain disease. So you see, like this one, it now also comes down to you. It will also be a matter of choice, because we are not encouraging you to discriminate anyone based on their genetic information, because we don't choose to be born with a genetic condition. It just happens, and you don't choose to have a risk with the genetic condition. It just happens. But what we are trying to say is, now that you are aware, okay, I am predisposed to this condition, or if I marry this person, or maybe I as the individual have this genetic predisposition, or I have like this medical condition that is genetically related, and maybe my partner does not have it, or I am considering marrying this person with this genetic predisposition. So I think it now comes down to you as an individual to make the decision. Will I be able, do I understand this genetic predisposition well, and am I able to handle it, in terms of, will I be able to like bear the consequences that comes into going into this joint marriages? So I would just like to stop it at that and not comment any further, but I think that it actually does depend on the individual choice. And in terms of the baby, if you realize like you are a pregnant woman and then you realize your baby will be born with a certain condition, are you then allowed to abort that baby? Well, clinically speaking, you should be allowed to, like, you should be given that right to make that decision, but it also depends on your country, like what laws are there governing this one. So that's why we're actually saying that in Africa at the moment, genomics knowledge is very new. It's still a baby. We're still, the baby is still learning to crawl. We've not walked yet, but we need to like establish clear ethical and legal frameworks and social frameworks that will enable us to manage these situations in a better way, even including paternity testing. Like I'm seeing someone had posted on the chats before, the issue of paternity testing, because that is also genetic information that we are providing. Because what if you come to get your to see if this is your son, and then in the course of doing that sequencing, we realize that okay, maybe the child is not yours, but could possibly be your brother's. So you see, now me as the geneticist, I'll remain with this question: should I tell him that, you see, this child is not yours, but it's the brother's, or should I just tell this person that this child is not yours, basically, and leave it at that? So there are a lot of ethical issues that are being raised by genomics, and thank you so much, Ms. Wangare, for raising that. I hope you've been answered, or if you would like to make an additional comment, this is your chance.
Yes, that was very well said by all of you. I'm learning a lot. Thank you.
All right, thank you so much, Ms. Wangare, for raising that issue. So I would like to take it back to our panelists. Ms. Neema, would you kindly explain to us the importance of having this genetic test, despite the many issues that are arising as a result of this genetic information that is being provided out here?
Okay, thank you again, Ms. Rissy, for this opportunity. As we saw earlier, Ms. Rissy, she captured so many applications of genomics earlier on, but maybe at this point again, I will choose to talk from a precision medicine perspective. Yeah, it's because it's a field that is fascinating me, where all this issue of tailor-making treatment according to a person's genetic, according to a person's genetic makeup, So it's really a fascinating field to me, so I'll choose to talk on the precision medicine perspective. So with genomics, with our sequences, we covered earlier what sequencing is all about. So we are able now to know variants. So we are able to know, first of all, first of all, I'm dealing with I'll just give you some bit of pros and maybe some cons of why we should sequence our genome. So it's through this sequencing that we are able now to identify maybe susceptibility of diseases and maybe correct and maybe or maybe put some intervention measures towards that. So maybe I can give you a case example of, maybe just imagine there's a person X, there's Mr. X and Mr. Y. All of them, they have malaria, okay? So they go to the hospital and they're given an anti-malarial drug, but it doesn't work for person X, but it works for person Y, okay? Yeah, it doesn't work at all for person X. He still has the malaria, but for person X, after a week, he's healed and is going about his businesses. So this is why we said it's because variability says, genetic variability says we can also be different in the way we metabolize or respond to treatment or drugs. Okay, yeah, that is what variability tells us. We captured earlier on that we are 1% different. So it's captured too in this, that we are different in the way we metabolize drugs. So Mr. X, again here, because the drug is not working, he goes to the doctor, and maybe the doctor asks to sequence their genome. And after sequencing, they find out that it's because of a variation on a certain position on the gene that metabolizes this anti-malarial drug. So that is why it doesn't work for Mr. X. He still has the malaria. Okay. So now through this information, the anti-malarial drug can be designed again for Mr. X. So maybe by reducing or increasing the dosage so that it works. Then after that, now, yeah, it's able to work. So this is now what we call pharmacogenomics in our field. Okay. Pharmacogenomics, where treatment is treatment of drugs or a drug is tailor-made for you according to your genetic makeup. So yeah, through your sequencing results, you are able to know that certain drugs may not work well for you, and maybe we may need to give you other drugs, that are related to that drug, or maybe play around with the dosage of that particular drug, maybe by increasing the dosage or reducing the dosage, so that it works. And this is because we want to avoid what we call adverse drug reactions. Okay, adverse drug reactions. Yeah, because it's usually postulated that many people are hospitalized more because of the adverse drug reactions, other than maybe the disease itself. Okay, so I don't know, adverse drug reactions. Yeah, I mean the 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 side effects of the drugs. Okay, so many people are usually hospitalized because of these adverse drug reactions more than the disease itself. So now that is pharmacogenomics. You are able to tailor make treatment for you so that some drugs can work for you. So it's really coming up on maybe cancer drugs and maybe cancer drugs, maybe HIV, antiretrovirals. Yeah, so it's really coming up. Maybe another point I can touch on is on nutrigenomics, where maybe certain variants, or in this field we call them SNPs. Yeah, so certain SNPs are associated with certain genetic, say maybe obesity. By knowing through the sequence of a person, by knowing that this person has those SNPs, we can. So by knowing that this person has those SNPs that can endanger this person in getting that genetic disorder, let's say obesity, so we can be able to tailor make again nutrition for this person, okay, so that we can correct or avoid those disorders. So if we know that maybe you will be susceptible to obesity, so you can start taking nutritive measures, like, yeah, you should avoid taking these kinds of foods and take these kinds of foods, yeah, so that you can be able to avoid that disorder. So that is called nutrigenomics. And another area that excites me all about this sequencing is newborn screening. Okay, this is mainly happening right now in the US, so where newborns, okay, newborn infants are screened for possible disorders in their genome that may not be evident at birth. Okay, so the infant's risk. Sorry. So the infant's risk to these disorders is identified early enough. Okay, and intervention is provided that will alter the cause of the disorder and prevent its clinical manifestation. How beautiful is that? So I hope that will reach Africa soon, where we are able to identify from that infant stage that this infant who will grow into an adult later on will be susceptible to these disorders, and from that stage you are able to contain, or you're able to alter we are able to alter the cause of that disorder. So we are able to curb it from that so you're able to curb that disorder from that young age, yeah, and prevent its clinical manifestation. So that is so beautiful about sequencing these genomes. You are able to identify all this information and take correction measures for our susceptibility to diseases, or maybe design drugs, because maybe some drugs may not be working for us. So that is why I find it so interesting. But however, maybe with all these advantages or beautiful stuff about sequencing, maybe the main challenge here, what we usually have is in the finding, is the issue of what we call incidental findings. So incidental findings are those findings, for example, the clinician is testing for a particular disorder, is analyzing your genome to test for a particular disorder, but in the course of analyzing your genome, you find out there are so many other there are so many other variants that can make you susceptible to so many other disorders. So yeah, so this is so in the course yeah, okay, so as I've captured, in the course of sequencing, you will find other variants that this person is susceptible to. So this is a major challenge of what we call these incidental findings. And since this field is limited, we may not be able to identify if these variants are of any clinical significance at that moment. So that is why we need more people to get sequenced. Okay, yeah, we need a lot of data. So many people need to get sequenced so that we can have enough data and be able to identify that to identify variants of clinical significance through our genomes. Okay, so and you should not be worried about data here. When we find data, it's usually identified, then deposited in databases. I captured that you are able to compare genomes, okay, through sequencing. So we are able to compare and identify these variants of clinical significance. Yeah, but the advantage, another advantage, however, is your genome is only sequenced once. Okay, you don't need to be going every now and then to the hospital to your clinician, sorry, to be having your genome sequenced. You just sequence your genome once, and that's also in future when other variants are identified that maybe might put you at risk. You can always be you can always be informed. Okay, yeah, so your genome is just sequenced once, so you should not worry about that. So, and this is according to the US NIH 2030 bold predictions that is working towards having our information, our genome information just right on your phone. So any variant that is identified, you can easily get information that, yeah, I'm susceptible to this, and yeah, I can take preventive measures towards that. So that is why I really find sequencing as an important yeah, as a very important venture to, yeah, to just do, and why it is, yeah, it's just so important and it's just so fascinating to me. Yeah, thank you.
Thank you so much, Ms. Neema, for that detailed explanation of how much how much we can get from genomics information, in the aspect that we don't just get to identify disease risks, but we can also be able to see if maybe I really like the part where you are talking about our response to drugs. So I would know whether if I take this medication, will it function well for me or not. That is very, very interesting. And also where you are talking about nutrition, that is also interesting. And one, Mr. Mwangi had also sent a question on that, saying that mapping what people eat is mapping what we should eat is going to be an interesting part. If we talk to venture capitalists, it would make a lot of sense to finance people. I don't know if you agree with him, but you can let us know. And also other issues like when we are sequencing your genome and we find other information that maybe we were not looking for, and we want to see, should we tell you this, should we keep it from you? That is also interesting. So Miss Glory, would you like to add a little onto what she has told us?
Okay, thank you, Ms. Makokha, for this opportunity. I think Ms. Hadara has already mentioned most of it, but I would just like to mention, and also at the same time, like, answer a question in the chat box asking if would one identify ancestral migration route and the country of origin from genome sequencing. So that's another interesting thing about our genome. It can tell us our ancestral root. So this is because there are variants, or should I say population-specific variants, unique to a particular populations. Like in Africa now, there are some variants that are unique to the African population. So if we find those variants in someone's genome, we can also immediately just presume that yes, this person has an African ancestry. So, and also we can use our genome to help us identify, like Miss Rissy has mentioned previously, our predisposing variants. So these predisposing variants are more like they show us if we are susceptible to particular diseases. So they just show us like our risk of developing particular disease. And this is not, when I say risk, it's not really definite that you actually develop this disease. It's just telling us that the presence of these variants shows like we have a higher probability of developing that disease. So what this can help us do is that it can help us adjust our lifestyle. So, okay, let me just pause to say that generally, when we talk about diseases or developing a particular disease, it's mostly influenced by our genes or environmental factors or interaction between our genes and environmental factors. So let me use an example like we've used previously in diabetes. So let's say a person has a particular risk of developing diabetes, and maybe the person lives a normal, a very healthy lifestyle, maybe exercise regularly, it has a controlled diet and all these other environmental factors. So having that, even with having the risk in the variant, it will not definitely, like the person might never even develop diabetes because of the lifestyle of the person. So what this is basically just telling us is we don't have to wait until we have particular diseases, or we have maybe particular serious diseases. We can have our genome sequence and we can know which disease we are predisposed to or we have high risk of developing, and we can make adjustments to our lifestyle or other factors that can be changed, that can be modified, because we can't change our genes, but we can change other predisposing factors that influences us developing those diseases, and this can help us lower the risk of developing a disease, and sometimes we might even end up not developing the disease at all. So if I will just sum it up, it's just basically empowering us. So I would say genome sequencing is empowering us to make informed decisions towards bettering our life and improving our health. So also we can talk about our family. I'm sure we've raised these issues before. So our genome doesn't just affect us, it affects our families as well. Because even though we are healthy right now, we might be also carrying specific variants in our genome that can be passed to our offspring. So having our genome sequence can help us know if we are carriers of these variants, and also make us, like I've said, help us make informed decisions. So these decisions would also be guided by our clinicians, our psychologists, and other team of people that will work together with us. Because I have to say this, in terms of genome sequencing, we are not alone. We will not be left alone to handle this, because the handling, knowing that we have a risk of developing Alzheimer's, is not going to sit well with even me. So I just want us to know that we are not alone. So there are going to be a team of people that will be with us to help us handle this information, and will help us make informed decisions that will help us to be able to live better and healthier lives. So I just want to make the example like we've used of sickle cell. So we know sickle cell to be a very serious disease, and people that have carried the S gene are carriers of this cell. So it means that if in choosing their spouses, if they marry someone who are also carriers, they have a very high probability of giving birth to children with this disease. So, but you see, sickle cell is just one of these many diseases that are out there. So there might be carriers of many other diseases that we don't even know about. And I think it's a good idea for us to actually know about them and make decisions, proactive decisions towards our life. So thank you.
So thank you so much, Ms. Glory, for that elaborate explanation on, and also for answering that question from Wangari. And maybe to add on to that question, yes, indeed, Wangari, you can be able to trace your ancestral heritage. There's a case of an individual who grew up in an orphanage and wanted to find out who the father was or where they grew up, and they were able to use genomic knowledge to be able to trace the father and even the family, and he was able to actually get to meet the father. And from what Miss Glory has just spoken about when it comes to this being able to, like, wouldn't you want to know if I'm going to be sick so I can prepare? Like that person who wanted to know if they can still eat their chicken, wouldn't you want to know if maybe in the next 10 15 years, let's say, you want to be able to eat that chicken, so you can see, do I want to eat this chicken more? I'm not encouraging you people to eat chicken, but it's just a thought. But or would you as a person want to say, okay, let me stay away from this kind of food so that I can be able to live for long? Because we still say these are just predispositions. It is not like a sure case that yes, if you have this variant, you will definitely develop this disease. No, we are just telling you you have a chance of developing this disease, and we still need more data from you individuals, you as a person. When this question ever poses up in your life at some point, would you want to have your genome sequenced? If you allow that information to be done, then we can be able to add this information to a database where other individuals also have this, and we can be able to tell, to like find out more information. Like we also need like individuals like the bioinformaticians, the data analysis. Like there's a lot of biological data that is available, but we have a little understanding of all that information because we don't have the expertise yet. The expertise is limited. We have few individuals working on this data, so we need more of you to actually join us in this train and be able to sequence more of the data, be able to interpret that data and make a more response about it. And also on the issue that she mentioned about sickle cell anemia, like September is actually Sickle Cell Anemia Awareness Month, and those who know me at a personal level know that sickle cell is something that is very close to my heart. So I would just like to comment that yes, indeed, it is a bit brutal to one end to say that okay, you need to know your genotype, but if you take like a country like Nigeria where the prevalence is very, very high, like sickle cell disease, like knowing your genotype, if you have the S gene or not, is a very, very important marriage question, like a courtship question, because we don't want you to, like, you get mad, like this information is already available. We don't want you to get to a situation whereby you already have the S gene, you've married someone with the S gene, you did not test, and now the child has the disease and you're complaining, oh guys, can you please come and help me? Like, I know we should actually empathize with you. It is understandable. It's the human nature. But if you can avoid it altogether, why not find options? If you really must marry this individual, there are other options that are open whereby you can be able to get your child. You can be able to get a child who does not have the disease, because as much as we are trying to reduce the amount of births of this disorder, genetics can help us with that, but genetics is also helping us to find a cure. So why don't we just help to reduce the number of births as at now, because genetics is not yet giving us a solution, and then when genetic does give us a solution, then you can go ahead as a young adult and get married to that individual that you want to get married to. So that would be my comment on that. And I also saw another, and also about heritage, the company had mentioned earlier, 23andMe. It can tell you your ancestry. So actually it's online and it does this globally. So if you are really curious about whether you are 10% African, are you really African? Are you truly African? How sure are you? Maybe your genomics will tell us something else. And also an interesting thing about that platform, you could be able to identify some relatives you don't even know that they existed. Maybe you have a brother in the UK and you don't know. Wouldn't that be something that you'd be interested in finding out? And so I'm just saying, I'm not really imposing this on anyone or encouraging anyone, but it's something to think and ponder about. And then I'm also seeing, I've also seen a question from Miss Marie, I think it was earlier, where she was asking, how can we translate this genomic information, these research findings, from the bench to the bedside, like moving it from the lab and taking it into the clinical, I'm assuming by bedside you mean clinical setup. Maybe she can elaborate on that, and what are some of the challenges. So Ms. Neema, would you give us a comment on that one?
Okay, thank you, Ms. Marie, for your question. I think that.
Kindly unmute your audio. Thank you.
Oh, thank you. Okay, thank you, Ms. Marie, for your question. So I think basically here I can take it like we need more of science communication. Okay, we need a way of how we can communicate from the researcher in the lab to the clinician in the hospital on how to communicate these findings to maybe now to the mass to the patients. Okay, so I think basically here we will base on science communication. Science communication here should be a key on how we can communicate with each other, from the researcher to the clinician in the laboratory to the patients or the masses out there. How to make them to make it known to them about this useful information. Because one thing I can give an example is how okay, we are usually like for example, when you go to a pharmacy, your prescription is written. You're even a patient is usually not able to read that information. It's only the pharmacists who can be able to read that information, or the clinicians. So I think here we should make science communication clear from all these aspects, from the research world to the clinic to the clinicians and to the patients or the masses out there. Yeah, that's my contribution.
Thank you so much, Ms. Neema. Ms. Glory, would you like to add on how we can make this information move away from the lab to the bedside of the patient? How can the patient directly benefit from this information?
I think Ms. Neema has already said it all, because communication is key. Because what good will it be to have to know that maybe you can't no, this particular drug genetically cannot work for you, when even your doctor doesn't know that and he's still prescribing that same drug to you. So what good would that knowledge be to you? So I think that is why it's important for communication and collaboration, if I can say that also, because the clinicians have to work hand in hand with the genomic scientists. And we know we've mentioned this all before, and I'll still mention it again. Genomic science is an interdisciplinary field, so there are a lot of people we are aiming to work with. We work with the clinicians, we work with everybody around us that will help to ensure that this knowledge is passed to the right person at the right time, because it also has to be time. Because you see a patient, like for example, now let me use the statistics that has been done. They said most of the times people don't go to the hospital, like statistics in the US, people end up in the hospital not because of a disease, but actually the adverse effect of the drug had on the person's disease. And we call this year's adverse drug reaction, and shortened for ADR. And in the US, they said ADR is like the sixth most leading cause of death. So that kind of information is quite alarming, and it's obvious, because if you look at it genetically, we can understand, because there are some drugs that need some enzymes. If I may go a bit into this area, there are some drugs that needs to be, let me use the word metabolized. It means, when we take a drug, it needs to be removed from our system. So there are enzymes that are responsible for that. So what happens to people that don't even have these enzymes? Because based on our genetic studies, we've seen people that lack these enzymes, or these enzymes has reduced function because of variants, like we've mentioned. So what happens to these people? It means when they take this drug, the drug will either have no effect on them, or they might end up with serious adverse reaction. So we, knowing that as genomic science is not just, as scientists is not just enough, we need to we have to have a channel where we can pass this information to the clinicians, so that the clinicians are the ones that will sit in front of a patient and prescribe the drug. So they need to know, so that they can look at the patient and be able to interpret their genetic results also, and be able to give recommendation or make better clinical decisions based on genetic information. Thank you.
So thank you so much, Ms. Glory, for that elaborate discussion on how we can be able to make this information available to other individuals from their lab to the hospital. I would also like to add that there's this model that is being tried currently in Germany, whereby we are trying to change like the traditional way in which you just go, the meeting with you and your doctor is just between you and your doctor, and we want to add like you need to have a genetic scientist in that meeting to be able to explain this genetic finding to you, and we also need the genetic counselor, so you guys are not left out if you have any counselors. Wangare, I know you're one. So if this is something that you might consider working with us, so that you can be able to handle the psychology part of this information, because for us we will just tell you this is it, but we don't know how to explain to the individual that okay, now that you have found this information, what will happen? That one we cannot handle it. So these efforts in genomics, it requires a multidisciplinary approach, and as Africa we are not really left behind. We just have a few gaps that we need to fill and to be able to get it. So I think at this point we would like to thank you all for coming with us, unless there's someone who has a really, really burning question and they're feeling like we are locking them out. This would be your chance. You can either raise your hand or you can just unmute yourself and speak. So anybody? All right, so then I'm assuming that every individual is satisfied up to this extent. So I want to take this opportunity to thank ADA Africa for giving us this platform to be able to talk about genomics, and I hope that to some extent you've been, at least you've had a bit of knowledge, such that when this question is bound to be posed to you 15 years from now, 10 years from now, or maybe 8 years from now, you would be able to actually make a bit of an informed decision as to whether you want to have your genome sequenced, or you would be like, no, I'm not curious, I'm not interested, it's fine. You guys can stay with your information. That is also allowed, because these issues, it is not something that we have to impose to you, like we will come and capture you and force you to sequence your genome. No, it is something that you are actually given a choice as an individual. Do you want to sequence or not? And then you say yes, we will sequence. If you say no, it was also fine, and we appreciate it. But in terms of improving the research field and improving our knowledge, we would really appreciate if each of you, maybe you go back and research. You can also get in touch with us if you need more explanations, if you do not understand some aspects, to be able to work with you in terms of making this decision. So thank you so much. I don't know if Wangare, you have something to add before we sum up?
Mine is just to say we are so honored by you, our very own Journal Club member, Rissy Makokha, because of your wonderful facilitation. And we are also really honored for our guests to join us and give us quite an elaborate understanding. I had many questions and all of them are answered, which is amazing. You've really delivered it in a simple way. So I see you're also experts in science communication. And I just want to also thank all of us who joined in as participants. Thank you for this space. And I'd like to encourage each one of us, aside from the main Journal Club group, if you are a guest, you haven't joined us, please do so. And you could also join a subgroup where you can actually create deeper conversations in your subject expertise area. So some of us belong to some subgroups, and these are all just community-led. Whoever in ADA Africa, we believe in developing research leaders, and we can see today Rissy is definitely a research leader. And so we want each one of you to also belong, not just to the general group where we're talking all matters research, but we also want you to belong where you can actually speak to the subjects. So even if you joined and you're not in genomics, I'm sure you understood much, and you are greatly assisted with all the answers. I still see more questions coming. And Doris is asking, do we have a place we can consult for diet? I think it's after I asked about the ancestry one. And there's a lot of thank yous coming in. PK saying it was a very huge learning curve, quite intuitive. And from now on, for me, my next move is to find a lab to have my genome sequenced. So maybe you could share your contacts, Rissy, again. I know you had shared them on the slide, so that those of us who would like to reach you for more information, at least now we know we have an expert in the house. We can consult on those matters. You could type it in the chat, or you could share your slide again so that we can reach you. And this conversation doesn't end here. We definitely have recorded it, and we will be availing it to those of us who did not make it. So on another note, we have a few other announcements. We need you to do our shut up and write session. This is an individual study time that you allocate. if you would like to get done with whatever you're working on for your research. We're a community who believe in being there for each other. So please join the group of cheerleaders and just, take on your shut up and write individual study session anytime and let us know how that goes. So nothing else from me. So I just would like to wish everybody a wonderful day. And back to you, Rissy, if you have any closing comments.
Thank you so much, Wangari. I think we've summed it all. If you have any further question, I'm not going to disable the link for the Slido. You can still send in your questions there and we can discuss this further. If you'd like to reach me also, I'm on LinkedIn as Rissy Makokha, and you can also communicate on Twitter as Rissy Makokha. And if you are in AIDA Africa, I'm a member, so you can just send your questions there. We can also communicate from there. So thank you so much for joining us today, and we're looking forward to having more conversations in this line. So send us your topics, things that you are curious about. We'll be able to try and answer them to the best of our ability. Thank you so much, and I'd like to call it a day from here.
Okay, thank you. And goodbye, everyone.
Yeah, goodbye.
So you can leave at your own pleasure.